We are honored to have Dr. Art Atkinson give one of the initial lectures of this course. Dr. Atkinson founded the NIH's course on Principle of Clinical Pharmacology almost 20 years ago. Dr. Atkinson received his undergraduate degree in chemistry from Harvard College in 1959 and his medical degree from Cornell University in 1963. Following residency at Mass General Hospital, he was a clinical associate in the Laboratory of Clinical Investigation here at the NIH's NIAID. He received his postdoctoral training in clinical pharmacology at the University of Cincinnati. In 1970, he moved to Northwestern University to start the clinical pharmacology program there. While at Northwestern, he and his colleagues set up the first U.S. hospital laboratory devoted to general therapeutic drug monitoring. In 1994, Dr. Atkinson joined Upjohn as a vice president for clinical development and medical affairs. Following the Upjohn-Pharmacia merger, he joined the Center for Drug Development Science at Georgetown University. In 1998, he returned to the NIH as a senior advisor in clinical pharmacology to the director of the NIH's Clinical Center. Dr. Atkinson has received numerous honors and awards over the years. I know you'll enjoy today's presentation.
Thank you very much, Dr. Fig. My topic today will be the compartmental analysis of drug distribution, and by that I mean the post-absorptive transfer of drug from one location in the body to another. There are a number of ways in which drug distribution is analyzed in pharmacokinetics. The first is a non-compartmental analysis in which there's curves are fit to the data, and this can be used to calculate parameters like the area under the curve, the clearance, the distribution volume, the Cmax, and the Tmax. What we'll focus on today is the second of these approaches, the compartmental analysis, where the parameters of a pharmacokinetic model are actually fit to the data. And usually these are fairly simple models with, say, 1 to 3 compartments, as opposed to the increasingly popular physiological models, which are much more extensive and actually represent a chemical engineering approach to the problem. Each organ is assigned its own compartment, and the model parameters are found from the physiological literature. I'm an agnostic about these modeling approaches and believe that the choice of model should depend on what the intended purpose of your analysis is. But today we'll focus primarily on the physiological basis of multi-compartment pharmacokinetic models. And we're indebted to a physiologist at the University of Uppsala, Torsten Tierell, who many years ago developed the first multi-compartmental model. He's regarded, therefore, as the father of both compartmental modeling and physiologically based pharmacokinetics. And this is a picture of one of his models, and what you can see here is a central compartment which represents the intravascular space. There's distribution into tissues here, elimination by various organs. Essentially, he was working with a 2-compartmental model. Now, what do these compartments mean? If we look at a classic drug like digoxin, you can see here that the distribution volume of 536 liters doesn't really correspond to what we can recognize as a physiological compartment. But we'll go into that in a few minutes and start with the conventional view of these physiological compartments that consist of first the intravascular space, the interstitial fluid space, and the intracellular fluid space. Well, most drugs don't have distribution volumes that correspond exactly to those compartments, but there are some compounds that do have a correspondence. And the classic marker for the extracellular fluid space is inulin. And proteins and other macromolecules also distribute in the extracellular fluid space, as do the neuromuscular blocking drugs, and initially the aminoglycoside antibiotics. There are a number of markers for total body water: urea, the caffeine that you have in your coffee or Coke, ethyl alcohol, and some people have used antipyrine as a marker, even though it does exhibit some protein binding. So how do we rationalize most of the distribution volume estimates that we find? There are really two factors that I think are most important to consider. The first is binding to plasma proteins. And we'll take thyroxine and theophylline as examples. And then there's the tissue binding or partitioning of a vast array of other compounds. So the first thing I would like to disabuse you of is the concept that plasma proteins are only distributed in the intravascular space. In fact, these binding proteins distribute throughout extracellular fluid space. So essentially, we have two main compartments to think about: extracellular fluid space and intracellular water when we think of distribution volumes. And one can mathematically analyze and predict the effects of protein binding on distribution volume with this fairly simple equation. What you see here for thyroxine, which has a protein binding of 99.97%, is that its total distribution volume corresponds with the expected value for extracellular fluid space. On the other hand, theophylline is a drug chemically very similar to caffeine. If it didn't bind to plasma proteins, its distribution volume, like caffeine, would be total body water. But because of the protein binding, its distribution volume is less. And here we can see that it's somewhat less than 50% protein bound, and so its distribution volume is less than one would expect for total body water. Now, this kind of approach allows us to understand some of the effects of physiological change on drug distribution. Let's take the example of the pregnant woman. In the third trimester, not only is the protein binding of theophylline decreased, but the fluid space estimates for extracellular fluid and total body water are increased. And so you can see, if we take all these factors into consideration, we come up with estimates for distribution volume that agree quite closely rather to what we can measure experimentally. Now, what about drugs that distribute extensively into tissues? Here we have to add another factor, phi, to our equation, and I will call this the ratio of tissue to plasma drug concentration. And generally speaking, this factor is correlated with the octanol water partition coefficient for the various compound in question. And this is a rough graph that shows this relationship. So let's get back to the example of digoxin. We know that the fraction free is 0.75, and we can back calculate from the observed distribution volume that has to have very high partitioning into tissues, a factor of phi of 20.4, and here's experimental data. The binding also includes binding to sodium, potassium, ATPase. See, every tissue in the body here has much higher concentrations of digoxin than does the concentration in blood. So how do we analyze experimental data using compartmental models? Well, the first question is, how many compartments in the model? A lot of people agonize about it, but my approach is rather simple. The number of exponential phases in the plasma level versus time curve will determine the number of compartments. And so here we have a bi-exponential equation. That means we're going to wind up with a two-compartment model. There are four parameters in this equation, two coefficients and two exponents. Four parameters there means that we can identify four model parameters. And here I've highlighted two rate distribution rate constants and elimination rate constant and the central compartment. But we could also calculate the two distribution volumes for the two compartments and two clearances. And the total distribution volume in the compartmental system is simply the sum of the two compartment volumes. And this now, to add to the confusion, gives you a third way of calculating distribution volume. We've already learned earlier in the course about VD extrap and VD area. This is the third way of calculating distribution volumes, and that obviously adds to the confusion. Now, the primary parameters of the system are not the rate constants. They're the distribution and elimination parameters are the clearances. And here we have, for our model, an elimination clearance. It's simply, as you know, the product of the elimination rate constant and the central compartment volume. There is a clearance also that describes the rate of drug distribution, and that is calculated in an analogous way to the elimination clearance. And like the elimination clearance, it's a volume-independent parameter that characterizes the rate of analyte transfer between compartments of the kinetic model. And this parameter has been really pretty much ignored in pharmacokinetic studies. We'll try to explain a little bit more about it as we go on. Let's go back, though, to our conventional view of physiologic spaces. And what I've shown you here is a three-compartment model, a catenary model. Comes from the Latin for chain. You can see the three compartments are linked together in this horizontal way. And these first two compartments, intravascular space and interstitial fluid space, are combined to form what we call the extracellular fluid space. Traditionally, we think of extracellular fluid spaces as having two compartments. And that understanding is based on this type of study. Here's a study done by Mario Gaudino way back in 1949, before he had computers to help him. You can see his experimental lines and his theoretical lines and his experimental data points after both a prolonged infusion and after bolus injections of inulin. And what I want to call your attention to is this region of his curves. You can see here, after the bolus injection, the data points are below his theoretical line. After the prolonged infusion, the data points are above. And this suggests that he's missed a compartment. And so these days, when we think of inulin distribution, we really have to think of a three-compartment model with a more rapidly equilibrating compartment and a slow equilibrating interstitial fluid compartment. In other words, interstitial fluid is kinetically heterogeneous. And the basis of this lies in the anatomy of capillary beds. The splanchnic bed has very large holes in it, and so solutes can pass into the splanchnic tissues much more rapidly than in the somatic compartment, where we have continuous capillaries. And here's a scanning electron micrograph of hepatic sinusoids. These are called fenestrae. I'm a sailor, so they look more like portholes to me, but they're big holes. And you can get big molecules going through these holes very rapidly. On the other hand, here's a high-power electron micrograph of the interendothelial cell capillary junction. And you can see how much narrower this is, certainly in this blow-up. And that retards the passage of hydrophilic solutes through these capillaries. There's a brilliant study done many years ago by Bob Sherwin and his colleagues at both Yale and NIH, in fact. And they studied the kinetics of insulin, a molecule about the same size as inulin, but obviously one that has a pharmacologic effect. And the pharmacologic effect of insulin that we're interested in is its ability to lower blood sugar by increasing skeletal muscle uptake of glucose. This study was done not only as a kinetic study, but as a study performed with a euglycemic glucose clamp. In other words, the investigators infused intravenous glucose so as to maintain constant blood sugars, even though insulin was acting to increase glucose uptake by skeletal muscle. And here's the slow equilibrating compartment in their 3-compartment model, and you can see it parallels skeletal muscle uptake of glucose, if you will. So this is one very rare example where the pharmacodynamic compartment actually is a pharmacokinetic compartment. And so for that reason, it's of a particular interest. Urea also can be described with a 3-compartment model. And the central compartment, again, if the urea is given rapidly intravenously, is intravascular space. So let's look now at a revised model of physiologic spaces. We have kinetic heterogeneity for the extracellular fluid space, and that also affects urea distribution into total body water. In both cases, the transcapillary exchange is the rate-limiting step, and that's the process that we would like to analyze in terms of understanding what makes up the transcapillary, the intercompartmental clearance of these compounds. There are actually 3 parameters that need to be considered: capillary blood flow, which we'll designate by Q, the capillary permeability coefficient, which we'll call P, and the capillary surface area. And this gentleman, Eugene Renken, who was former chairman of physiology at UC Davis, was the first to apply this equation to the transcapillary clearance of solutes. It's a rather simple equation, and it can be adapted to study the intercompartmental clearance of inulin and urea. The challenge, as you can see, is we have experimentally one term, a clearance term, and we'd like to calculate 2 terms, a permeability coefficient, surface area product, and a flow term. In order to do that, we have to study urea and inulin simultaneously. In both cases, transcapillary exchange is the basis for the intercompartmental clearance. And we need 3 equations. So the first 2 equations are rearranged versions of the Renken equation. And the last equation simply assumes that the transcapillary exchange of inulin and urea will be the ratio of those permeability coefficients. The surface area products will be the same as the ratio of their free water diffusion coefficients, which you measure in vitro. And if we do that and simultaneously measure cardiac output, we now can compare our flow estimates for the 2 compartments in our model with independently measured cardiac output. And you can see here the correspondence is fairly close. So obviously most pharmacokinetic studies are not performed with this level of detail, but in certain circumstances it can be illustrative to have this amount of detail in our analysis. In the subsequent lecture, we'll talk about how we can use these models to understand physiologic changes that occur during hemodialysis that obviously have an impact on hemodialysis pharmacokinetics. And they also have a bearing on analyzing why patients with reduced cardiac output might not absorb drugs as well as patients whose cardiac status is better. Now, you'll have a subsequent lecture on drug absorption, and all these various factors will be considered. But the one factor that's hardest for us to get our hands around in clinical studies, at least, is the importance of splanchnic blood flow. Well, remember our model. The fast equilibrating compartment is the distribution of drug in many of our models to splanchnic tissues. And here we're looking at a drug, N-acetylprocainamide, where again the central compartment is intravascular space. We have fast and slow peripheral compartments. And you can see that if the fast intercompartmental clearance is less than 1.1, there's a reduction in bioavailability. Now, obviously, what we're really trying to get our hands on is splanchnic blood flow. And we can use, in this case, fast intercompartmental clearance perhaps as a surrogate for that. Well, what are the mechanisms for transcapillary exchange of a variety of compounds? We've talked so far about polar uncharged compounds, inulin and urea. Here we've said the transfer should be proportional to the free water diffusion coefficient. But there are many small molecules, some of them polar, that have a slower transfer rate than we would predict from their free water diffusion coefficient. Some of these are highly charged, and others have properties that cause them probably to interact with those pores in capillary walls that we've been talking about. And certainly the quaternary compounds have a major interaction. In other cases, the transfer rate is much faster than we'd predict. For example, the studies with the antiarrhythmic drug ibutylide, the intercompartmental clearance to the peripheral compartments from the intravascular space is some of those clearances adds up to what we'd expect for cardiac output. And lipid-soluble compounds of a variety of compounds act that way. The other perplexing thing is that theophylline actually goes faster across capillaries than we would predict from its free water diffusion coefficient. And this is a very unusual study in which theophylline pharmacokinetics were studied in dogs simultaneously with urea and inulin, and we measured the cardiac output as well. And what you can see here for theophylline is that the intercompartmental clearances for theophylline are very close to blood flow calculated from the urea and inulin kinetics. And why should this be? Well, theophylline obviously is bigger than urea. It has a corrected Stokes-Einstein radius that's bigger. It should go more slowly. The free water diffusion coefficient is less, and yet it crosses capillary walls faster than urea. So the assumption is there is a facilitated transport carrier, as yet to be identified, that is responsible for this rapid transfer. Now let's continue this line of thought by considering some of the clinical implications of drug distribution kinetics. First of all, there are some drugs that are given rapidly by intravenous injection are quite toxic. Theophylline, again, is a case in point. In the late 30s, it was found to be a valuable drug in treating patients, asthmatic patients with status asthmaticus. But several years later, it was noted to have both fatal arrhythmic toxic effects and central nervous system cardiorespiratory toxicity. It's interesting. It's been a puzzle to me why people wait for 3 cases before they write a paper, but that seems to be the magic rule. Even in 1971, it was known that the injection of this drug intravenously, even over a 3 to 5 minute period, resulted in 60% of drug-related cardiac arrests in the Los Angeles County shock ward. So people correctly surmised fairly early on that speed of injection was important in this toxicity, and I will say that the reason speed of injection is important is that speed of distribution of this drug is extremely rapid. And of course, the current recommendation is to slow it up and give the theophylline over a 20 to 40 minute period. Well, conversely, distribution can delay the onset of drug action of a number of compounds. We've seen previously how the delayed distribution of insulin to skeletal muscle delays its onset of pharmacologic action. And let's go back to digoxin again. You can see that there's a slow buildup of digoxin concentrations in the heart, and consequently a slow onset of the therapeutic myocardial effects. On the other hand, the peripheral vascular system reacts to the high intravascular concentrations that are immediately obtained, and that causes vasoconstriction. And the bottom line of this story is if you have a patient with congestive heart failure and pulmonary edema, that if you begin therapy with a cardiac glycoside, this delayed effect means that the initial effect you'll observe is a reduction of cardiac output, and the patient will become worse. Now, another consequence of this disparity is that the digoxin, if ingested in a suicide attempt, say, can distribute very rapidly to the central nervous medullary vomiting center. And so the result of this action is that the patient will eliminate most of the ingested dose before it has a chance to kill them by working on their heart. You can think of this as sort of a protective mechanism that has been designed by the ultimate designer, I guess, to protect us to some extent from eating poisonous plants like foxglove. Well, distribution also terminates the action of many drugs given after a bolus dose. And perhaps the one that you've observed is the fact that if you have somebody with a ventricular arrhythmia, say a VTAC, and you give them a bolus injection of lidocaine, after 8 to 10 minutes, the arrhythmia comes back. And that's not because the patient has eliminated the lidocaine, it's because the lidocaine has redistributed from the intravascular space to rapidly equilibrating tissues, and the metabolic elimination is a relatively slower process. There are certain special cases that have been designated flip-flop kinetics, and the poster child for this type of a drug is gentamicin. All the aminoglycosides, though, will demonstrate this. And currently we give this drug in a daily dose, and you see the plasma concentrations fall after each dose. And that decrease in concentration actually reflects the elimination of the drug. If you measure concentrations after the drug dosing is stopped, though, you see a prolonged distribution phase. So here we have drug elimination phase preceding drug distribution phase. That's the very unusual. The distribution phase actually represents distribution of gentamicin primarily from extracellular fluid space to the kidneys. And Coburn and Shentag and their colleagues at SUNY Buffalo have shown that patients who show nephrotoxicity actually are accumulating more of the gentamicin in their kidneys. They can actually measure the amount of gentamicin in the kidneys in post-mortem exams and account for almost all of the gentamicin that is distributed to the peripheral compartment. I'll finally talk about something that I'll call pseudo-dose dependency, and something that causes the inaccurate estimate of pharmacokinetic parameters. One of the most toxic non-cancer drugs that we use is dilantin or phenytoin, and you can see here that the plasma levels increase much more rapidly than the dose is increased. And so this does not satisfy a criteria that we call dose proportionality. And so let me give you an example from the real world of drug development. The Upjohn company was developing a drug, linezolid or Zyvox, and the phase 1 study, pharmacokinetic data, showed that after a 25 milligram dose, the AUC was 1.32 micrograms hour per milliliter. When a 100 milligram dose was given, a 4-time increase in dose, the AUC increased 13 times. And this data was presented to the senior management at Upjohn Laboratories by the pharmacokinetics group, and there's incentive in the industry to stop the development of a drug that isn't going to work out as early as possible. So the development of this antibiotic was about to be stopped when, fortunately, the pharmacokinetic group showed their plasma level versus time curves. What you see is after the 25 milligram dose, their assay wasn't sensitive enough to transcribe the entire plasma level versus time curve. So they only had this truncated curve. And so, in fact, that threw the estimates off. The drug did show dose proportionality, but let me show you what the pharmacokinetic estimates would have been had they just worked with these data. You can see here that with the inadequate description of the plasma levels versus time curve, you had an underestimation of distribution volume and an overestimation of clearance. Now, the current practical significance of this is that we're being confronted with a whole bunch of wonderful drugs that happen to be rather large molecules. And what I'd like you to do when you examine pharmacokinetic data for these drugs is to compare the distribution volume spaces with those of inulin. What you can see here is that, generally speaking, the central compartment, the intravascular space estimates, are generally pretty much in line with what you'd expect. Factor 9 is a bit unusual in many respects, seems to bind to something. But for most of these drugs, the total distribution volume is much smaller than the estimate of extracellular fluid space. And that means not only that the distribution volume estimate is incorrect and too low, but the clearance estimate in the published papers is incorrect and too high. So I want you to try to remember that as something perhaps you've learned from this particular lecture. And I want to thank you for listening, and I hope it's been enjoyable and instructive. Thank you.
I am sad to report that since the filming of this presentation, Dr. Art Atkinson has passed away. Dr. Atkinson was one of the founders of the NIH Principles of Clinical Pharmacology course and was a true pioneer. His leadership in the field of clinical pharmacology will be missed.